In this section, we're going to take a deeper dive into the carrier-mediated transport proteins and look at some special characteristics of what makes them work well. And then we'll go a little further onto glucose transporters. So if you'll remember, there are two types of transporter proteins. There are channel proteins, which are non-specific, and then these carrier-mediated proteins, which are specific for the types of molecules they'll be transporting. And remember, they're going to be doing the large molecules, such as glucose, and polar molecules, which may be things like amino acids or proteins. One of these carrier-mediated proteins are called our GLUT or glucose transporters. So there's many different types. I'm just showing you here 4 different types of what we call GLUT transporters. Remember, they're specific for the type of molecule. This is one of the big things about these. They're specific for the type of molecule that's being transported. And these different versions of this same transporter exist in different parts of our body. So GLUT1 transporters are going to be transporting glucose in and out of our red blood cells, across the blood-brain barrier, and a little bit in our heart. The GLUT2 transporters are going to be important in our liver and pancreas and small intestine. So the GLUT2 are for more digestive purposes. GLUT3, think nervous system for the third type of transporter. So your brain, primarily all that your brain uses for energy is glucose. You learned that a little bit in the last chapter. So these GLUT3 transporters are going to be very important there. And then in our body, for skeletal muscle and adipose tissue, these GLUT4 transporters are the ones that are active there. These guys are the ones that are going to be involved with maintaining our insulin. So they're taking the blood sugar out of our bloodstream and storing it in either the muscle or the adipose tissue, or being used quickly around the heart, because the heart's always using glucose for aerobic energy. So GLUT4, circle and underline that. These are going to be the important ones for us for muscle and fat. So if we talked about specificity of molecules, again, thinking about, here's that ion channel. It's not taking every molecule that's going around there that will just move through it like the carrier channels. It's only taking the ones that fit through there. That's the specificity. But they can only move so fast. They can only get so many molecules through at once. And you can kind of see that from here, only a little bit at a time. So they have what's called saturation. And what that means is, as you increase the concentration of a molecule, you're going to increase the rate that it'll transport. So that kind of makes sense. The more molecules that you have down here, the faster they'll push through this little protein. But at some point, that protein will only be able to get through so many molecules at a time. You can think of it like kids going on a bus. There's a door on the bus. You can have a bunch of kids waiting to get on or to get off the bus, but only so many can fit through at a time. That's going to be your rate of transport. And it's what we call is a saturation, is our transport maximum, saying what our fastest rate of transport is going to be through that protein. So if you have an X, the rate of transport will be some level here. Now, we talked about competition as well. So some of these carrier-mediated transport proteins can accommodate a couple of different molecules. So let's say there's a molecule Y. Let's say that Y can go through that molecule at the same rate that X can. So it can either take X or Y, and they'll have the same saturation point. But if you combine to have X and Y's down here on the same side, and you try and get them through at the same time, if you look at either X and compare it to now, X's maximum rate of transport is going to be less than what it would have been if it was by itself, because it's competing with Y. So if they're both the same rate, then this would be about half of what it would be by itself. So that's what competition does. It's going to lower the rate of transport of any one of these molecules. Add them together, and X and Y would both be up here at the same point. So just to quickly review, in looking at facilitated diffusion using these carrier-mediated transporters, we know that they don't require ATP. They're going from high concentration to low concentration, down their concentration gradient. They're specific, so only certain molecules are going through across that plasma membrane. Some of these transporters can transport more than one substrate, and we call that competition. And here's the new thing. The transporters either always remain in the plasma membrane or they may be inserted as needed. So these proteins right here sometimes exist in the cell, and then when we need them to come to the surface, they'll come out here. And we're going to show you how that happens. So these glucose transporters that I talked about before are a perfect example of that. If this was in your muscle or an adipose cell, this would be a GLUT4 transporter. And you'll see that we've got these little transporters here, all stored within the cytoplasm. They're kind of attached to each other in this case, making a little ring of carrier proteins. And when we have insulin out here, the insulin is going to signal this cell to bring these transporters out to the plasma membrane. And then when they do that, they'll be available for all the glucose that's in there to pull the glucose into the cell. And when it's not needed, they'll come back into the cell. So now if you imagine that this is your plasma membrane, and this will be the inside of the cell, and this is the outside of the cell. When you eat something, so you have food, maybe an ice cream or a sandwich that's got glucose in it, that glucose gets into your blood. Well, it's going to be floating around your blood. It can't go into the cell until this GLUT transporter is here made available. It's not always available. What actually happens is this glucose in your blood is going to signal your pancreas to release insulin. That insulin then gets into your blood, and that insulin is going to settle on a receptor, an insulin receptor, and when it gets activated, it's going to cause changes within the cell, and we'll see this in the last video of this chapter, in which it causes changes in the cell. It brings those GLUT4 transporters that we saw in a little ring earlier up to the surface, and once they come up to the surface, then they can bring that glucose into the cell. And remember, this is a facilitated carrier-mediated transport, so it does not require energy. No ATP required here. It's just going down its concentration gradient. In the next video clip, we'll look at transporters that do require energy, and those are going to be called primary and secondary active transport. But this is facilitated passive.